All right, class, this is Dr. Anderson, and let's start talking about skeletal muscle. We are going to talk about some organelles and transporters, some of which you've already encountered under the neuron section, but we'll add some muscle proteins that are involved in signaling and involved in the contraction process. When we talked about neurons, we talked that neurons are really permeable to potassium. Skeletal muscles are also fairly permeable to chloride, and we want to think about why that might be. We also want to think about why muscle cells have T-tubules, and basically think about how we're going to excite the entire diameter of a muscle cell that might be 100 microns in diameter. Just for comparison, a red blood cell is 7 microns. So a muscle fiber could be 100 microns in diameter. So how are we going to accomplish that? As you may have known from your other coursework, the basic structure of a skeletal muscle fiber is the sarcomere. So we want to talk about all the parts of a sarcomere and how a sarcomere shortens. We want to talk about how muscle is activated via the neuromuscular junction and review the steps of synaptic transmission. And we also want to think about how the neuromuscular junction compares to a central nervous system synapse. We want to talk about something called excitation-contraction coupling, which is how does an action potential end up causing calcium release and contraction. So what are the molecular events that occur once we get calcium release and contraction and the use of ATP and all that sort of thing? What's happening to the parts of the sarcomere as it shortens, and how are we going to make this muscle cell relax? So this is a picture from your textbook. I don't have it in your regular PowerPoint because those of you that print your notes really don't need to be spending a lot of ink printing this beautiful picture, but I adore this picture. So this is a Z line, this is a Z line, and so this structure in between is a sarcomere. Notice there's some blue places, that's the I band. Notice there's this pink and yellow area, that's the A band. This is where the myosin is, this is where the actin is all by itself. There's an H band or an M line down the middle that kind of anchors the proteins in the middle. We also see some vesicular structures. Oh, here's a beautiful one right here. And there's some here, and there's some here. Those are membranes of the sarcoplasmic reticulum and the T-tubules. They come together to form something called a triad, and a triad is very important for that process of excitation-contraction coupling. So if we look at muscle under the light microscope, we have a cartoon down here, and here's a light microscope. You see this is the edge of a fiber, that's the edge of the fiber, and you see these beautiful striations. So skeletal muscle is called striated muscle. You also see that a muscle fiber, which is the same thing as a muscle cell, is multinucleated. It's got many nuclei, because it would be ridiculous for the messenger RNA from this particular nuclei to have to diffuse all the way down a cell. So there's sort of nuclear domains along the cell, and this nucleus is taking care of this part of the muscle, and this nucleus is taking care of this part of the muscle, so on and so forth. Muscle cells can be very long, and can be 50 to 100 microns across. So these are very large cells. They contain all sorts of membrane proteins. So let's think. Sodium, potassium, ATPase. Hopefully you remember that you have encountered this before. This is a primary active transporter found in just about every cell of the body. Its job is to pump sodium out of the cell, pump potassium into the cell and maintain asymmetric concentrations of sodium potassium, such that there's high potassium inside a cell and high sodium outside the cell. We have a sodium calcium exchanger. This is a secondary active transporter. So we spend energy using the sodium potassium ATPase to create a sodium gradient, and using that sodium gradient we can get calcium out of the cell to keep calcium low when we want the cell to relax. We have voltage-gated sodium channels and potassium channels. Again, we have encountered these proteins before. They are responsible for the depolarization and the repolarization phase of the action potential, respectively. We have voltage-gated calcium channels, and there's a slow, what's called a slow L-type calcium channel, and it just so happens that it binds a compound called dihydropyridine. So this protein, though it is a calcium channel, is also called the dihydropyridine receptor, and it's very important for excitation-contraction coupling. We have leak channels. So we have leak channels for sodium, not very many. We have lots of leak channels for potassium, quite a few, and as potassium leaks out of the cell, we generate a negative resting membrane potential. We also have leak channels for chloride, and we have quite a few of those. So if you, in combination, think about the action of these leak channels, the resting membrane potential is minus 80 millivolts. It's a little more negative than what we saw in a central nervous system neuron. So there are many layers of structure. So we start with a whole muscle that's attached tendon to tendon to bones. If we cut down through the middle of that muscle, the belly of the muscle, we can see that muscle has a lot of connective tissue. So we have bundles of cells that are wrapped in connective tissue. Those are called fascicles. Then we see some blood vessels and nerves that run in the connective tissue. And then the entire muscle is wrapped in connective tissue, and that connective tissue wrapping is very important for attaching the muscle to the tendons. If we pull out a single muscle fiber, we can see the muscle fiber has the striations, and within the muscle fiber are many, many, many myofibrils. And a myofibril then also shows this striping pattern, and it's within the myofibrils that we have the sarcomeres. So if we take one sarcomere and blow it up, we see we have the M line, the H zone, we have the A band where the myosin filaments are, we have the I band where only the actin is, we have some other we see some other proteins here that help stabilize the structure, and then we have the Z line. So myosin is a polymer of many, many molecules of myosin. They look like double-headed golf clubs. All the shafts of the golf clubs are together, and then the little myosin heads stick out in all directions. Actin is the thin filament with a couple of other regulatory proteins. So actin is like a double strand of pearls that's been twisted. There's a little ribbon of tropomyosin that goes through the double strand, and periodically there's a globular protein called troponin. Troponin is very important for the regulation of this whole thing that we call the contractile apparatus. Troponin is able to bind calcium, it's able to bind tropomyosin, and it's able to bind actin. So there's quite a bit of membrane organelle inside of muscle cell. So this gray part here is our plasma membrane. We also call that the sarcolemma. And notice periodically along the sarcolemma there are these little holes, and they're actually little tunnels. These tunnels go way down into the muscle. These tunnels are called T-tubules or transverse tubules. inside the transverse tubules is essentially extracellular fluid. And they surround muscle fibers, and then they're very, very close to another membrane system called the sarcoplasmic reticulum. So we have the SR, the T-tubule, and more SR. And so those are three membranous structures that together we call the triad. So there are going to be lots of voltage-gated sodium and potassium channels allowing an action potential to travel along the sarcolemma. Well, it turns out the T-tubules have voltage-gated sodium and potassium channels. That allows the action potential to go down through the T-tubule so that the entire diameter of the cell has been informed that an action potential is occurring, so that the entire muscle or the entire fiber can release calcium and the entire cell can contract. So I ask a question. Unlike neurons, skeletal muscle is highly permeable to chloride, and why is this beneficial? Well, the thing is, there's extracellular fluid in these T-tubules, and over many, many action potentials, there's going to be a buildup of potassium. What is going to prevent these cells from getting overexcitable? Well, this high chloride permeability. Chloride, remember, is a stabilizer. So if potassium builds up in the T-tubules and the cell gets kind of depolarized, well, chloride can enter the cell, return the cell to its more negative resting membrane potential, and electrically stabilize skeletal muscle. If we continue to look at the structure, out here in gray is our sarcolemma. In the purple are the T-tubules burrowing into the cell. Here's our sarcoplasmic reticulum. So there's an enlarged portion next to the T-tubule forming the triad, and then there's lots of stuff in between. What we're going to find is calcium is released at these terminal sacs, and the longitudinal portion is where a calcium ATP is that removes the calcium and returns it to the sarcoplasmic reticulum. We need lots of mitochondria in muscle, and they look kind of like little sausage-shaped things. Turns out in skeletal muscle the mitochondria form networks. They're very oddly shaped. They're just really well suited for muscle to provide that ATP for all the things that are going on in the muscle. We also see that our muscle fiber is filled with myofibrils. And then you can see on the end, the larger dots are myosin, the tinier dots are the thin filament actin, and they all are throughout the myofibril. And if you pull out one of these myofibrils, you nicely see the sarcomere with actin, the thin filament, and myosin, the thick filament, and they're overlapped a little bit. So how does the process of excitation-contraction coupling occur? And this is a beautiful picture to show this. This is the sarcolemma. An action potential is going to travel. I'm having trouble keeping my slides where I want them. An action potential is going to travel along the sarcolemma, make its way into the transverse tubule, and as the action potential travels along, it's going to meet a dihydropyridine receptor we see in this enlarged circle. The dihydropyridine receptor is a calcium channel, but its activity as an action potential is not really what we're interested in. We're interested in the fact that this protein is voltage gated. And because it's voltage gated, it has something in it called a dipole. A dipole is a negative and a positive charge that can be flipped by a different electrical environment. So when the dipole is flipped in this DHP, it causes the ryanodine receptor, or a calcium release channel, in the enlarged sacs of the sarcoplasmic reticulum to open. And we get large amounts of calcium released from the lateral sacs of the sarcoplasmic reticulum. That calcium diffuses through the cytoplasm around the contractile apparatus. It finds troponin. It binds to troponin, which changes shape, which pulls on tropomyosin, which reveals binding sites on actin. The myosin heads, which flip up and down all the time, but when the binding sites are revealed, myosin can bind to the actin binding site. Myosin does something called a power stroke, and that allows actin to slide along the myosin. And when you have lots of actin and myosin sliding and furthering their interdigitation, the sarcomere shortens. And when sarcomeres shorten, then myofibrils shorten. And when all the myofibrils are shortening, then the fiber shortens. And when lots of fibers are shortening, the whole muscle is shortening. And it's just unbelievable that this tiny cellular event is translated into this entire muscle shortening and pulling a load. Notice that in the longitudinal portion of the sarcoplasmic reticulum, there's a calcium ATPase. So calcium is pumped back into the sarcoplasmic reticulum, so that there's a gradient for calcium to leave. When calcium is pumped back into the SR, and calcium release stops, then calcium is removed from the cytoplasm, and that permits the contractile apparatus to relax. So here's a beautiful electron micrograph of a sarcomere. So there's the Z line with the I band, I as in light, A band as in dark. So this is where the myosin is. This is where only the actin is, and there's an area here where actin and myosin are overlapped. If we look at the cartoon, actin and myosin are a bit overlapped. There's some other muscle proteins, for example, titin is an interesting protein we find in the sarcomere that has a stabilizing effect. But here's our Z lines, and here's the M line, the H zone in the middle. Here's the amount of overlap. And when myosin starts to pull on actin, the overlap is going to increase, and that is the basis of the sliding filament theory. So here you see more relaxed sarcomeres. As they slide, you see the A band stays the same. The filaments themselves don't contract, they change their amount of overlap. And so you see the I band is going away as more myosin is overlapping with actin. So cross bridges occur between the myosin head and the actin, and actin and myosin bind, then myosin does a power stroke. And as myosin moves, if it's attached to actin, then actin moves as well. Again, actin has a binding site for myosin. However, much of the time, that site is not available because it's covered up by tropomyosin. So the position of tropomyosin is regulated by troponin. So notice this myosin head is labeled as energized. It's bound to ATP. ATP has been hydrolyzed, and the energy on that myosin head is just sitting there ready to go, but there's no actin to bind to. When calcium comes along, calcium binds to troponin, pulls tropomyosin out of the way. We have all these binding sites available, and now this energized myosin head can bind to actin, forming a crossbridge. When the phosphate and the energy from ATP is released and ADP remains, that allows that myosin head to ratchet. It pulls over, and it pulls actin with it. So when we think about muscle conditions, a very important pathological issue with muscular dystrophy, with muscle, is something called muscular dystrophy. And I absolutely have to talk about muscular dystrophy because at the University of Minnesota, the Department of IBP is sort of a powerhouse of muscular dystrophy research. So muscular dystrophy, it's a group of inherited disorders. it causes the muscle to be damaged. That results in muscle weakness. People lose their muscle function. Or they lose the ability to walk. It attacks the diaphragm. They lose. They go into respiratory failure. It also affects the heart. And actually a lot of progress has been made in the treatment of muscular dystrophy and the treatment of the pulmonary failure. And now what people die of is the cardiac failure, and that's a area of active research in our department. So many of the cases of muscular dystrophy are inherited. You may be aware of a type of muscular dystrophy called Duchenne muscular dystrophy. It's a X-linked disease, and what that means is there is a mutated protein on the X chromosome. If you're a girl, you have another X with another protein that can compensate for the loss of your of the mutated protein. If you're a boy and you have a mutated gene on your X chromosome, you have a Y chromosome that does not have a normal gene and cannot compensate. So boys tend to have Duchenne's more than girls. Girls can have it, but only in certain conditions. So there's a Becker muscular dystrophy, which is a little less severe than Duchenne's muscular dystrophy. There are other types, and many of them are genetic. So here's an interesting thing. So there were people that had muscular dystrophy, and then with molecular and genetic research, they realized the mutated protein was in a structure called a costamere, and the protein was named dystrophin. And essentially what happens is the contractile apparatus is connected to these costameres. And as the muscle contracts and relaxes, the costamere, and particularly dystrophin, acts as a shock absorber and basically helps alleviate stress to the membrane as this thing is contracting and stretching and contracting. Well, if your dystrophin is mutated, then you lose your shock absorber function, and then you damage the membrane. And as the membrane is damaged, then there are satellite cells that come in there and try to repair things, but you run out of satellite cells, and then the cells continue to get damaged. So people are looking at gene therapy. People are looking at your CRISPR. I can't think of the CRISPR thing where you try to correct a mutation defect. People are looking at blood supply. If you make sure the muscle gets a good blood supply, is it able to heal itself better? People are all over the university are working on this disease and making great strides. So you should be proud to be at the U of M where this great work is going on. All right. Here's an electron micrograph of the neuromuscular junction. So you have one alpha motor neuron with various branches, and the branches synapse on one muscle fiber. So a motor unit is an alpha motor neuron and all the muscle fibers it innervates. If we look at a cartoon, here's our myelinated axon, here's our axon terminus, here are vesicles of acetylcholine. In here we're going to have a voltage-gated calcium channel. So when an action potential travels down, we're going to release these vesicles. Acetylcholine is going to diffuse across the synaptic cleft and cause a graded potential at the postsynaptic membrane. If we reach threshold, which we typically do, then out here we'll find voltage-gated sodium and potassium channels, and the action potential will then be propagated along the sarcolemma. So here's another beautiful picture with all that detail. So once again, we have an action potential going down this myelinated axon. In the axon terminus, we have a voltage-gated calcium channel. Calcium comes in, that allows synaptogamma to facilitate the fusion and release of acetylcholine. Acetylcholine diffuses across the synaptic cleft, finds the acetylcholine receptor. This is a nicotinic receptor and it's ionotropic. Acetylcholine binds, this opens this beautiful channel, which is a cationic channel. Sodium comes in, calcium comes in, potassium goes out. But what ends up happening is we get this big graded potential called a end plate potential. When acetylcholine releases, it's destroyed by acetylcholinesterase. The byproducts are taken back up into the presynaptic cell. We're going to reach threshold. That depolarization then opens up our voltage-gated sodium channels, and then we can propagate the action potential along the sarcolemma. So the acetylcholine receptor, it's a large ligand-gated channel, nicotinic and ionotropic. And if we took a muscle and allowed the acetylcholine receptor to just sit open, the cell would stabilize at 0 degrees. So the reversal potential of the acetylcholine receptor channel is 0. It's kind of like an equilibrium potential. We leave these channels open and we see where the membrane potential stabilizes at. So it doesn't go up to positive 60, it goes to 0. So sodium is not the only ion going through this channel. It allows calcium to come in, but it also allows potassium to go out. So the reversal potential for this channel is between sodium and potassium's equilibrium potential. So there is a type of drug called succinylcholine. I'm sorry, I had trouble with that. Succinylcholine is a depolarizing neuromuscular blocker. So succinylcholine binds to the acetylcholine receptor, opens it, and keeps it open. That depolarizes the cell, keeps the cell at 0. Sodium channels get inactivated, they can't reactivate, so the cell can't have any more action potentials. Why does this elevate serum potassium? Because potassium is permeable to this channel. Potassium will exit the cell, and muscle cells have truckloads of potassium in them. So you can get a significant rise in serum potassium, and so you need to be aware of that, and maybe the nurse anesthesia students could share their perspectives on this when we talk about it in class. But you need to be aware of an increase of potassium that will occur with the use of succinylcholine. Another disease that affects muscle actually works on the neuromuscular junction. It's an autoimmune disease. It tends to happen more often in women. I don't know why that is, but people make antibodies to the acetylcholine receptor, and the antibodies destroy the channels or make them non-functional. Well, if you can't activate the neuromuscular junction, you're going to get muscle weakness. So a treatment of this condition is a drug called neostigmine. Neostigmine is an acetylcholinesterase inhibitor. It allows there to be higher amounts of acetylcholine in the synapse, increasing the probability of activating the synapse. Because it's an autoimmune disease, people either take immune suppressors or they can have a surgery called a thymectomy. So your thymus is a gland that's responsible for maturing T cells, and it seems to help the autoimmune aspect of the disease by removing the thymus to treat this disease. Another muscle disease that is important for nurse anesthetists to know about is malignant hyperthermia. This is a genetic disorder where halogenated anesthetics, so there's an anesthetic called halothane, and it's not used anymore, but really it wasn't that long ago. Okay, so maybe it was 20 years ago. That doesn't seem like that long ago to me, but anyway. Halothane was used on kids Uh, and it just it's wonderful that there are other alternatives for anesthesia, because halothane would inappropriately activate calcium release in people with malignant hyperthermia. Then, so here these people are in surgery, and they start getting intense contractions, and they're producing all kinds of heat, and they've got acidosis and lactic acidosis, and this is not a good thing to happen during surgery. Now the problem is human mutations are very wide and varied. Some families have mutations in the ryanodine receptor. Some families have mutations in the dihydropyridine receptor. And so because there's so many mutations and they're different from family to family, there is no genetic test for humans. So you can take a muscle biopsy, send it to a lab. They can add caffeine or they can add halothane. You get these contractures. That's a positive test. Now, it doesn't necessarily mean you have malignant hyperthermia. You could have some other muscle disease, but it'd be something you'd want to be aware of. There is a treatment. You give somebody sodium dantrolene, which blocks the dihydropyridine receptor and blocks the calcium release. So it is very, very effective, but you have to recognize an episode is occurring, and you have to know where the sodium dantrolene is, and you have to know how to mix it up and that kind of thing. So it may never happen to a practitioner, but if it does, you got to know what to do. It's very, very treatable, but if it's not treated, the person will most definitely die. So when people go in the hospital and they're having surgery, they have to be asked, have you ever had anyone have surgery and have something go wrong? Or have you ever had anybody die because they got anesthesia? You're trying to ask if maybe malignant hyperthermia runs in the person's family.